I'm going to welcome our speaker tonight, Danny Miller. I met Danny at the Pacific Northwest NAMA camp and saw his lecture and thought it was something you guys would really like. Um, Danny is a member of the Puget Sound Mycological Society and is the education chair for that club. He helps design and teach the curriculum for the club's mycology classes. He is also the club librarian, an ID committee coordinator, and an emergency poisoning point person for King County, Washington poison control. Like a lot of us, once you sort of are in, you kind of get really in and pick up a lot of different side jobs because there's a lot to do once you're kind of in the mushroom world. Um, Danny also belongs to the Pacific Northwest Key Council, which is a group of amateur and professional mycologists, and is co-author with Ian Gibson of the free Pacific Northwest ID program, MycoMatch. And we can, if you go to the website and see Danny's bio, there's some links to these things. He has a big interest in taxonomy and figuring out where all of the mushrooms fit into the fungal tree of life, and is currently trying to collect DNA of every Pacific Northwest species to determine which still need names. And then you can follow the results at Danny's DNA Discoveries, which I am going to post into the chat, the link. And I'm going to invite you all to also put questions in the chat. And most of them will probably save towards the end, unless they're really key to the timing of the presentation. So, Danny, thank you so much for joining us. We're really excited to have you here.
Thank you. I really appreciate the invite. I'm here today to talk about some of the really, really cool discoveries and theories that we have now about the evolution of mushrooms that's coming up from the study of DNA. It's been really, really fascinating. The project that I'm doing to try and sequence all of the mushrooms of the Pacific Northwest and catalog them. Whenever you start any project, biodiversity always ends up being much bigger than you expect. And that's the same thing here. We thought maybe we had a few thousand mushrooms, maybe 2,000 or 4,000 mushrooms here total. And we've had 10 to 20,000 DNA sequences made. And one out of every 10 to 20 sequences is still coming up brand new as something we've never seen before. So there's a whole lot of biodiversity out there that is making this a giant, well, it's just going to be a multi-generational project is what it's going to be. So I'm here to talk about some of the cool things that people have discovered when looking at the DNA sequences of mushrooms, and because it kind of tells a fascinating story that isn't really what you might think. So as humans, we love to classify things. It's not just enough to name all the mushrooms. You have like a million names on a very long piece of paper. That's very confusing. That really doesn't get you anywhere. We want to put things in boxes. We want to take all the ones that look almost the same and make a genus out of them and say, here's all the ones in that genus. They're very similar. And then a family for the ones that are sort of kind of related. And we do this sort of tree classification. Here's an example of a tree here that sort of shows how we think they're all related to each other. Since the dawn of time, we've been doing this kind of thing to make trees to figure them out. Now, in the beginning, before the microscope, before everything else came along, if you wanted to classify your mushrooms, all you could do was look at it. And if two mushrooms look kind of the same, you'd say, well, maybe they're related to each other. If they look really, really different, you'd say, let's put them in a different part of the tree. That's all you could do. Elias Fries was the first person to really systematically apply the Linnaean system of genus, species, family to mushrooms. And he started writing books around 1821 and classified them. So he's the guy who sort of decided how it was going to work. And he said, well, everything that has gills under the cap is probably related to each other. They probably are. That's what most people think of when they think of a mushroom. They think of it has a cap, it has a stem, and you look underneath and you have some gills. And the more you get into a club and the more you look around, you realize the diversity of how many different shapes there really are. But he decided that that probably meant they were all closely related to each other. And then the boletes, like the porcini, which have that sort of spongy pored surface underneath, they were probably all related to each other. So the number one had a way to determine what sort of family or genus your mushroom is in, was what was under the cap and what it looked like. Now, there's so many gill mushrooms that the next step he decided was going to be the spore color. So for all of you who are upset that identifying a mushroom is so difficult that you can't just look at it, you have to actually wait 4 hours and put it on a piece of paper and figure out what colors of the spore to get anywhere as to identifying it, you can blame him. Now, to be fair, it's really not his fault. It really is an informative thing to know. The spore color turns out to be an important piece of information, so you can't blame him. But it started way back in 1821 that if you wanted to identify a guild mushroom, the first question you would be asked is, what's the spore color? And that's kind of stuck. So some tough decisions had to be made when you did it this way. So he had Agaricus, which was the genus for all guild mushrooms at the time. Not only did they have gills, but they were often growing on the ground. They weren't always attached to wood. Some of them were on the ground, and they were soft. You could actually eat them. And then on the far right-hand side here, the polypores or conch were hard and tough. If you tried to eat one, you'd break your teeth on it. And they're attached to wood, and they have a sponge, that spongy pore surface underneath. And those were, he called the polypores. And then they discovered this weird little guy in the middle. It's a tough, hard conch growing on the side of the tree. But then you look underneath, and it has gills. So what do you do with this guy? How do you classify it? Is it a guild mushroom that happened to evolve to be harder and tougher and lose its stem? Or is it a conch that just happened to evolve gills underneath instead of pores? Well, they had the rule. The rule said the number one thing to consider is what's under the cap. So it was considered a guild mushroom at first. Were they right? Nobody knew for sure. Well, spoiler alert, turns out it was wrong. Now, the next step, sort of in the evolution of classifying mushrooms, is the invention of the microscope, because some really fascinating things were found. If you look under the microscope at most guild mushrooms, you'll see the spores are these sort of ellipses, usually like 10 microns by 6 microns or so in that ratio, maybe a little bigger, maybe a little smaller. And besides the spores, you see, as you see in the middle picture, these long, stringy fibers in it. Mushrooms are very fibery, right? You can sort of peel stringy bits off of them. And that's usually what you saw. And then somebody put a russula under the microscope, and it looked completely different. The spores are perfectly round, and they have these little warts on them that turn black in iodine. And not only that, but most of the rest of the cells that you see are also round. See all these like big sort of elliptical round things everywhere, not a lot of long stringy fibers. If you've ever thrown a russula against a tree, which is a lot more fun than it sounds, it'll kind of disintegrate into circular bits. It won't leave long stringy bits behind. It just kind of like goes poof and vanishes. Can be really fun because of the cell structure. So this was a clue that something was very different about russula, and that maybe it can be more important what's microscopically too small to see than anything that was large enough for a human to notice with their naked eye. That could be more important. And it turns out that russulas are nowhere near a tree of most other guild mushrooms. They evolve completely separately in a very, very distantly away. Here's another example, the gild bolete. If you've ever come across this guy in the forest and you thought you find like a Sewillis or some kind of a bolete, because then you turn it upside down and it's got gills instead of a sponge. It even turns blue on the gills wherever you touch it. It's got that bright yellow surface underneath the pores. It's got that kind of like suede like brown top. And people were like, wow, this is like a bolete, but it has gills. And the microscope confirmed that, because most boletes, like this sort of normal bolete on the left, they have really long, thin spores, sometimes 3 times longer than they are wide, a very different shape. And you can see the gill bolete has the elongated, not quite as much, but very, very long spores too. So this is where they started to realize, okay, maybe the system of saying that everything with gills under it is more closely related and all the things with sponges are more closely related might not be true. So it is true. The gill bolete really belongs in the family with the boletes. Russula is this third group altogether. And so they try to classify every mushroom. They relax the rules if, that Fries came up with at the beginning, if the microscope showed that similarly looking mushrooms really were quite different than each other and vice versa. But did they get it right? Is there even any such thing as a right answer? Is this all going to be a matter of opinion forever? Well, DNA has come along, and most people are now pretty much agree that this is the sort of the final answer of which mushrooms are, of how to classify them. It has been sort of officially decided that if two mushrooms share the same great grandparent, that means that they are very closely related. And that's the way to do it. That probably really does have the most meaning. You can imagine if you're wondering if a mushroom is poisonous or edible or not. You can look at its closest relatives and it share many of the compounds, and that might tell you the role that they play in ecology might be similar. If you're looking for mushrooms that have health benefits, or you might look for the ones most closely genetically related to them. So now we have a way, finally, of making the tree of life for fungi and everything else that can give us the most meaning of how they're really related to each other. So here's an example tree here of the fungal kingdom, Basidios and the Ascos. You might know that we're sort of splitting up the mushrooms into Basidios and Ascos. Most of the things you find are Basidios, the most famous Ascos being morels. Those are probably the most famous ones. And if you look, there's a couple, and the stars, I put stars next to the ones next to the fungi that have fruiting bodies big enough to see. They're not just powdery molds. They actually produce mushrooms as a fruiting body. And most of them are in the Basidios and Ascos, but there's actually a few that aren't, which is kind of interesting. It's not true that all mushrooms you might find would be either a Basidio or an Asco. Let's show what the difference is under the microscope. Once again, the microscope tells you right away whether you have which sort of half of the fungal kingdom something is in, where to just know that by the naked eye would be very, very difficult to come up with the rules to figure that out. But the microscope shows it right away. On the left is what you see for a Basidio. They're kind of like chandeliers with the spores hanging off them like lights. And ascos are these big straws, like pea shooters filled with them. And a top opens up and like those spores will kind of shoot out the top. And these are the structures that are found in the sexual stage of the mushroom's growth. So most fungi species are just powdery molds or mildews and things that don't produce big fruiting bodies. And for instance, this penicillin growing on a bread here. There are, as I said, a few types of mushrooms that are neither Basidios or Ascos, which is kind of cool to see. On the left here, these Endogonae, these yellow blobs, are neither. These little needles, and you can see there's little like pinheads at the end of each needle, which is a spore sac where all the spores are. And that Snellus, that is a fungus that's neither a Basidio or an Asco. And then you have things that people think might be mushrooms, like these slime molds that just are neither. They're a completely separate kingdom, but they're mycologists feel sorry for them because not a lot of people study them. So it's usually a mycologist that ends up doing the studying of the slime molds. So there's so many different shapes have evolved. And the original sort of classification system assumed that all of the mushrooms that had the same basic shape would probably be related to each other. And we'll investigate how that as you know from being in a club and getting exposure to many more types of mushrooms than the general public does, mushrooms come in all kinds of cool shapes and sizes. And this is the table of contents from the MycoMatch program that was mentioned at the beginning of the lecture. So here is a tree of the Basidios. And next to each branch, I've put the picture of which shape mushroom might grow in that tree. So the original theory was you'd see one picture in each branch. All the corals would probably be close together. The guild mushrooms would be together. The boletes would be together. The ones with teeth under the spine would spines under the cap would be together. And the truth is so complicated. I don't expect anyone to really know or care what these names are or which ones go with which. The thing that I want you to get out of this slide is how many different shapes are on each branch. There's sort of no rhyme or reason to what the mushroom looks like to whether or not they're related to each other. These shapes have evolved back and forth over and over again. It was one of the most fascinating results of DNA sequence. So let's look at the bottom half of the tree. You can see jelly fungi are some of the most primitive ones that probably evolved early on, and closing corals and puffballs. There were no guild mushrooms yet. The top half of the tree, the sort of more derived branches, is where you start to see guild mushrooms and even a whole lot more variety there. So I'm going to take you through what I've come up with as a theory of the evolution of Basidios, not just my theory, but putting together a bunch of things that that we've learned from DNA sequencing to sort of walk you through what the evolution of fungi might have looked like. So in the beginning, we have crusts. Crusts are one of the more primitive forms, easiest to evolve. They're just a flat surface growing on their food source. They're decaying the wood, and that's where they are. And they, unlike the powdery molds, they have some texture to them. You can sort of peel off a piece where you can't do that to molds like penicillin. It's kind of just dust. And so it's textured, it's coherent, and this is a basal form. You will find crusts in practically every single branch of meat. So one of the first kind of evolutionary things that probably happened is the crusts start to bend away from the wood, because even just bending away and like 1 inch or so gives you a little advantage in the wind that your spores can get caught and have a better chance of flying further away. So that's one thing that happened. Another thing is the textures, like this phlebia down in the bottom left corner. The flat surfaces became wrinkled, more and more wrinkled, because the more wrinkles there are, the more spores you can stuff into every nook and cranny to just be able to create many more spores than you otherwise would. Mathematically, that's called maximizing the surface area to volume ratio, which just basically means put as many spores in as small a place as you can. And stems help too. As a mushroom would grow on a stem coming out of a branch, then that would give it that extra maybe inch or two to be able to get a little further, get caught by the wind more, and be able to spread further. So, as I said, crusts are found in almost every branch of the tree. Just looking at a crust, you really have no idea what it's related to, what else it is. And their identification is kind of to be despaired of. You need at least a microscope, perhaps DNA sequencer, to figure out what it is. And I think there's probably a special place in heaven for people who devote their lives to studying and identifying the crusts. So let's walk our way through here, the tree, and kind of look at some of the important events of evolution in mushrooms. The very, very basal branch are a bunch of jellies. Tremella, witch's butter, that actually in the most basal Basidio branch. So here's some witch's butter and other tremella typings. So the first jellies to evolve, they're just kind of blobs of jelly, are actually parasitic. Now, being a parasite can be a quick and easy short-term strategy for surviving, but it's not a very good long-term strategy necessary to kill your host, and then you can't get any more food if you do that. So, but it's interesting that these most primitive branch are parasites. So they're not actually digesting the wood. The witch's butter is eating a crust like Asterium, as you can see in the bottom left photo, some Asterium crust growing on that trunk. It's parasitic on that mushroom, eating the Asterium, and the Asterium is a crust on the wood eating the wood. So once again, microscope can tell you so many important things that you wouldn't tell by the naked eye. If you look under the microscope to see the exact shape of the bacilla or the shape of those chandelier type things, it's number D here, which I've circled. Very, very sort of tall, tall, tall columns for the spores to grow on. And they're tall because they have to escape the sort of gooey jelly to be able to get outside that mass. And so if you see a basidia shaped like that, you know you have the kind of jelly that is a tremella. The next branch of the tree is also jelly fungi, the mycetes, which are more common on conifers than the tremellas, I think are at least around here, are more commonly found on hardwoods. And it's interesting because now you have some more complicated shapes. Some of the jelly fungi in the Dacrocetes, like the one on the top right here, it's almost got a cap and stem. You have like a shoehorn shape there. They're not just blobs anymore. So the shapes are more complicated, which would make sense because it's a derived line. And not only that, but they're not parasites. They are saprophytic that are eating and decaying wood. And they will not harm, usually not harm a living tree. They wait until the tree is dead, and then they move in and they help digest it and decompose it. So it's a helpful, not a harmful ecology and more advanced shapes. And my favorite kind of basidio, the tuning fork basidia, number B here, kind of looks like a tuning fork, is what you'll see under a microscope. You'll know right away that you have Dacromycetes type of jelly. Now, if you and I were to try and eat a piece of wood, we wouldn't be able to get any nutrition out of it. It's not digestible. That wood is just too tough and hard. But wood is made up of two main components, which is that hard brown woody stuff, and cellulose, which is like this white goo that kind of binds it together. You can't really see it's there, but it is. And that gooey stuff that's in there somewhere is much easier to digest. So that's what the first mushrooms ate in the wood when they were digesting. They digest the easy to digest cellulose, and they leave behind all of the blocks of the really, really hard lignin. So if you're in a forest, you see this cubically rotted, mostly decomposed log that's broken up into chunks, it means the mushrooms that were in there were brown rot mushrooms. The color is named after what's left behind. The brown stuff was left behind. The white stuff was eaten. So you know that you've got a mushroom that's a brown rot fungus. And that's what they did. Now we move up a couple of branches and we see another branch that's full of jelly fungi, the auriculariales, which is the tree ears and things like we have in the top left corner here. They're also saprobes, decaying the wood, and they have even more advanced shapes like ears. They look like shoehorns here, or one of the coolest ones, the tooth jelly that actually has the lines hanging down underneath the cap, like one of the tooth mushrooms, except it's a little piece of jello. And some of them now are white rot. I'll talk about that in a minute. First of all, let's take a look at what their basidia look like under the scope with all these sort of horizontal lines transecting the basidium. Is how you'll know you have one of the auriculariales under the microscope. So this is around the time when the auriculariales branched off from the rest of the basidiomycetes. This is the time, or the Basidiomycota. This is around the time that mushrooms evolved an enzyme to be able to digest wood, the lignin in the wood, which is really hard to do. Termites can do it because they eat wood. Mushrooms can, not a lot of other things that I know have the ability to do that. So here you can kind of see what that white cellulose looks like because all of the brown wood has been eaten and the white stuff has been left behind. So if you've ever come and seen this sort of gooey mess in a forest, that's what's going on. It was a white rot fungus that left the white stuff behind. And that's the kind of mushroom you found. That's the kind of mushroom that would have inhabited that log. What's really interesting is almost all, if not all mushrooms I know are one or the other. They'll digest the cellulose or the lignin, but they can't do both. They're each specialized. So the next super cool thing that mushroom evolved the ability to do was to leave their food source. Up until now, I've been only talking about jelly mushrooms that are attached to wood because they're physically attached to their food source. So mushrooms have then eventually developed the ability to live in a symbiotic relationship with the trees, that mycorrhizal relationship. So the threads, the invisible hyphae of the mycelium that are growing throughout the ground would attach themselves to the tree roots and get in a symbiotic relationship with the tree. So as we all learned in like around fifth grade, uh, tree plants are green because they have chlorophyll. And chlorophyll is this magic chemical that can turn sunlight into sugar. So the plants make their own food, which is why they're stuck in one place. You and I can go wandering around and shopping for food, but trees are kind of stuck where they are. So they can't go out and catch food. So they make their own sugar. But what I never thought about in grade five is that can't be the whole story because that is how they get sugar. But that would be like you and me on an all Twinkie diet, which would not be healthy at all. So the question is, where do the trees get their vitamins and minerals from? Everybody assumed, well, there's vitamins and minerals in the ground, and the tree roots are pulling the vitamins and minerals out. Turns out the tree roots are very poor at doing that. But mushroom mycelium, very, very good at doing that. So the arrangement was that the mushrooms pull the vitamins and minerals and things out of the soil, and they give them to the tree, and they say, "Hey, I'll trade you if you'll give me some sugar, because that's really hard to get calories." And they do that. So now that they could do that, the the mycelium would just grow to a random spot on the forest floor and sprout a mushroom anywhere far away from its food source. So you no longer see mushrooms that are attached to pieces of wood. You see them on the ground. And not every mushroom that is on the ground is mycorrhizal. Many of them are just decaying the leaves or the blades of grass or the things that are there. But definitely all the mycorrhizal mushrooms that you'll see are going to be the ones that are just growing out of a random spot on the forest floor. They did an experiment that probably several times to prove this, where they had a tree growing in a sterile lab where they didn't let any mushroom species in the soil. And the tree grew up really sickly, like that Charlie Brown Christmas tree and the Charlie Brown Christmas special. That's a really sad looking thing. And then they sprinkled some spores in and let the mushrooms grow in the soil, and the tree perked right up, like on the right, sort of pretty much this how important mushrooms are to the health of everything in the forest. Most trees and shrubs are kind of relying on mycorrhizal relationships of some kind with fungi in order to get some of their nutrients. Okay, so let the diversity begin. We're going to take a look at all the different shapes of the mushrooms and take a look at just how many places it's found, which is kind of interesting, and have a nice little fun review of the wild variety of mushrooms that we can find out there. Chanterelles, they kind of have these what I call speed bumps, veins underneath the cap, not quite gills, more like kind of speed bumps on the road. And we have the chanterelles like on the top, but also the false woolly chanterelle and the blue chanterelle on the bottom right are similar, but they're found in different parts of the tree. So on the bottom, I've put the number of orders of the basidios that you can find this shape in. So 4 different times at least, the chanterelle shape has evolved. They're not all related to each other. Clubs and corals. Clubs are pretty simple. They're just kind of just a straight line of club just grows out of the ground. Except the stinkhorns are really, really cool clubs. The ones that hatch out of eggs and they smell like poop to attract flies to spread their spores. Pretty clever reproduction strategy. Clubs and corals are found in 7 different orders. We have a cordyceps. For those of you watching season 2 of The Last of Us, that's a really prominent in our culture now, the zombie show that imagines that cordyceps can actually infect humans. So knock on wood, not yet. Corals, beautiful, beautiful coral fungi like the colorful ramarias and other coral-shaped things like Thelephora, the cauliflower mushroom kind of has this coral shape. 7 different places. This kind of makes sense because it doesn't seem like it would be terribly difficult for to evolve into a coral shape. It's just a branch club. You just grow a club and decide to branch into two or three, and then that branches into two or three, and then that branches into two or three, and then soon you have this really complicated shape. Tooth fungi, where you see those like spines or teeth hanging under the cap. Three different orders that you find those in. There's the hedgehog and the hawk's wings and other things that are found in three different orders. And here we have the little ice pick fungus growing out of a little pine cone, which is really, really cute. There's a toothed conch that has teeth underneath it instead of the regular pores. And the bear's head and other the sort of like icicle fungi, the toothed fungi, they're just blobs sitting on the tree. Polypores, the conchs, are found in four different orders. You have an artist conch here, you have the red-belted conch, a whole bunch of different ones. Boletes, now this is really interesting. Boletes and Sewillis, all of which are defined as they have that sponge underneath the cap, but they're soft enough to eat. You don't break your teeth on them like you do the conchs. A sponge where the sponge is removable and it's soft enough to eat only evolved in one order. This shape just evolved once and didn't happen again. I have no idea why, but that's really, really interesting to me. Birds' nests, which are so cool. They look like little tiny birds' nests. The spores are in packets of things that look like eggs. These are closely related to dark spored guild mushrooms, like agaricus, like the portobello, and the button mushroom. How did that happen? Not sure, but we do have some clues I'll talk about later on as to how this kind of thing can happen, how something so very different different looking might evolve and not too long for something completely different. Ascos have a whole bunch of other shapes that aren't typically found in the basidios and provide even more interesting diversity to look at. Morels, which knock on wood coming up soon, and false morels. Those are ascos and a really, really great shape. They're found basically in one class of ascos, mostly together. And the elven settles and what I call the brain mushroom on the right, kind of those Gyromitra false morels. Cups, cup fungi. The one in the middle, the orange peel cup, that kind of looks like you found a discarded orange peel and then you look closer and it's a mushroom. I've also made that mistake the other way around. I thought I found the mushroom and it was an actual discarded orange peel that was in the forest. So all of the large cups that are, say, more than an inch across, and all the small ones that are growing not on wood are in one class. And the small cups that are growing on wood and the earth tongues, which are kind of like clubs with a differentiated head. They're mostly these club-shaped things, but you can really sort of tell that the head is something different. They're found in a different class, which is interesting. And you can look under the microscope, although this is really hard to see, which class of ascos has to do with the little lid at the top of the tube and how the spores come out. There's either just a hole that it comes out, or it might have that opens and closes to let them out. And whether or not that's true tells you which class of asco you're in. And when I tried to think of how to make a key that actually talked about what class something was in genetically, I started to realize that most of the ones in this other class were the tiny cups that grew on wood, and the ones that grew on the ground or dung or sand were in the other class. So ecology turns out to be a really strong indicator of what things are related to each other. If they share an ecology, they're more likely to be more closely related than if they just look the same. Some flubs that are ascos. Here's the cordyceps again on the bottom left. The lobster mushroom, which is really cool. There's a whole class of ascos that have pimpled surfaces. If you look closely at the cordyceps here, you can see all the pimples on its head. And one great way of identifying a lobster mushroom is because there are other orange mushrooms out there that you might mistake it with, is the whole orange surface is covered in pimples. That's a really great way to identify it. And so this class of ascos only makes spores inside the pimples. There are no spores in between the pimples. So it doesn't make as many spores, but it has this advantage by having them inside this pimple. They can kind of like erupt out of a volcano and get this extra oomph and go further. So they kind of trade off the ability to eject further. So like quality over quantity of spores that they're making. And all of the ascos that are pimpled are indeed in one branch of the tree. That's something that tells you where to find it in the tree. So this is called convergent evolution when mushrooms look the same, but they happen to evolve independently, coincidentally, quote unquote, to look the same as other mushrooms when they're not really closely related. And it seems remarkable that so many of these shapes are found in so many different parts of the tree, that like corals evolve 7 different times at least, probably more, not just once. But if you think of only so many shapes can reproduce successfully, mushrooms will always find one of those shapes that works. And they evolve back and forth, finding a successful shape. And maybe it's not quite as crazy to think about them changing shape as we thought. Like if you have a gill mushroom, you turn it upside down and you pinch 2 gills together every like half an inch or so, you're kind of making a whole bunch of pores and holes in there. So maybe it's not as difficult or crazy as we thought to evolve back and forth between some of these shapes. So I found some really interesting things about spore colors and the evolution of spore color gill mushrooms, because everybody's always saying spore color because it's such an important question to answer when trying to identify a mushroom. We have here the very first gilled mushroom ever named, Agaricus campestris, the meadow mushroom. 1753 it got its name, so that's a pretty long time ago. White seems to be the ancestral color for spores. Most fungi, many fungi, or most of the ones that produce mushrooms, seem to have white or pale spores. There's like perhaps a reason to have to develop a color. Now, russulas have a really cool spore print color that other things don't. A yellow-orange spore print. And now this starts to make sense, because now that we've seen under the microscope, we realize that russulas are not closely related to other guild mushrooms. They're a distant branch all by themselves. They evolved under completely separate circumstances. So why not evolve a different spore color as than all the other mushrooms? So that kind of makes sense. And here's to show you how far apart they are. At the very top, the agaricales is where we find all of the is where we find the most of the guild mushrooms, and the russulas are way the heck down here, quite distantly related. Pink spores are cool. That, for whatever reason, I don't know why, evolved twice. The Entoloma family and the Pluteus, or deer mushroom family, evolved pink spores. And so that's kind of cool. If you have a pink spored mushroom, you have a much better hope of identifying what it is, because there's fewer of them. Yeah, and there's few typical differences in them. If the gills are attached or touching the stem and it's found on the ground, it's in the Entoloma family. If the gills do not actually touch the stem, they're free here. You can see like an ant could run in circles around the stem and not touch either the stem or the gills. And growing out of wood, touching wood instead of growing from the ground, you have a Pluteus family. So it's kind of cool that pink spored mushrooms are a little easier to identify than others. And the other cool thing is, under the microscope, entelomas evolve these like angular stop sign shaped spores that you don't see anywhere else. I have no idea why. Anybody knows, put it in the chat. But really, really cool, almost like little stop signs, angled spores. And electron microscope photographs on the right show you a little more detail of exactly all the ridges that are growing on those spores. So that's pretty cool. Now, all the dark spores colors, all the different shades of brown and black and purple brown, that evolved once. There's one branch of the guild mushrooms trees where the spores evolved some kind of dark color, and they're all together on that same branch, which is really interesting. It's a very large branch, but they're all together. and it's the most derived clade, one of the more recently split off groups of all the ones in the fungal kingdom. Now, while dark spores only evolve once, more than once, mushrooms have decided they didn't really need the color in the spores and have lost their spore pigment and gone back white spores. And Leucaria is one of them. These cool little Leucarias, which kind of almost look like waxy cap mushrooms, they're kind of like fake little wax mushrooms, really, really cool looking, have white spore prints, and they're right in the middle of the tree, surrounded by a bunch of dark spored mushrooms. Another group that lost their spore pigments are the Lepiotas, the Lepiota family. One genus was actually called Leucoagaricus, which is for white agaricus, because they look like portobellos, just like the agaricus, except instead of seeing the dark chocolate gills, you turn them upside down and they're pure white gills. And they look like they're basically portobello mushrooms that lost their spore print color. And that's kind of what they are. They're very close in the tree. Now, it may have been up to sort of 3 separate events that the different Lepiota type things lost their spore colors. But that's what happened. Now, some of the coolest mushroom spore colors, red and green spore print, only happened like once that I know of. There's a Melanophyllum that has red spores, and the chlorophyllum with green spores, one of the shaggy parasols that is very, very poisonous. A lot of the ones with white spores are edible, but the one with green spores are poisonous. This is the only example I know of commonly that you're going to see these colors. So where do these colors come from? Well, they're both in one of the Lepiota families, which means that it's not that they evolved red or green colors. They used to be, their ancestors used to be dark spored mushrooms with all of the colors in it. And whatever happened to make them lose all that pigment, one pigment got left behind. In Melanophyllum, the red stuck behind, and the chlorophyllum, the green stuck behind. So it's not that they evolved their green colors, is that they lost all of the colors except red or except green. So that's pretty cool. The kind of like if you have one of those old-fashioned TVs with you have the RGB input with the 3 wires, and like you accidentally knocked out 2 of the wires and the whole football game looks solid green because the 2 wires came out. That's my analogy anyway for what happened with these mushrooms. Now, most non-gill mushrooms have white spores. That's typically what you find. But for whatever reason, some of the gill boletes evolved dark spored colors. Pexus and Campanella and the gill bolete, these are all in the bolete order, and they have brown spores. And the Gomphidius and Croogomphus, the sort of slimy, hideous Gomphidius, and the sort of tent spike mushrooms have almost pitch black spores, and those are also gilled mushrooms that are in the bolete family. Not all bolete type gilled mushrooms have dark spores. Hygrophilopsis, one of the false chanterelles, has white spores. But it's interesting, and I don't know why some of these bolete relatives have developed spore pigments as well. One cool thing that came out of looking at mushroom DNA was the inky caps. It turns out that the mushrooms that deliquesce and turn to ink are not all related to each other. There's 2 families of them. There's the shaggy mane on the left, which is fairly closely related to Agaricus, the portobello type mushrooms. And all the others, like the alcohol inky that you can't have with alcohol, and many others, are closely related to Satharella. So this evolved twice. This is a really unusual skill and thing to evolve twice, to turn to ink. But they probably did it for 2 different reasons. The reason the shaggy mane probably evolved to turn to ink, have you ever got one and cut it open and looked at just how many gills it has? It's millions of gills. They're so close together, they're practically all touching. It fits like a million gills in there. And they are too close together. So the gills are so close together, when a spore falls or is ejected off of a gill, it hits the other gill. It doesn't have enough room to escape. So at the same time that it evolved accidentally, I'm personifying. I really like to personify this as if the mushrooms are people. They sort of accidentally evolved the gills to be too close. They might have gone extinct if they didn't have another strategy, and they happened to evolve at the same time the ability to turn to ink. So the cap just melts away, and the spores just kind of flow away in a stream of ink, and that's how they get spread. And I don't know why some Satharellas developed the ability to turn to ink. Perhaps I think somebody hypothesized it has to do with better for wind dispersal or something. I'm not sure. Um, but that was one of the interesting things that we learned, how there's two different groups of inky caps. So I've talked about a whole bunch of kinds of mushrooms. I've gone through them all. Why haven't I yet talked about puffballs and truffles? Did I just forget? Well, no. Truffles, very interesting. Very simple shape. They're just blobs. And you might think that truffles would have been one of the first things to evolve. You just have to be a blob. No complicated shape had to evolve. They seem very simple, but they're not. If you think about it, you find truffles in the ground, buried underground, not touching wood. So already that's a clue that they can't be that primitive a kind of mushroom because they're not attached to their food source. Most truffles are in a mycorrhizal relationship with trees. They're more advanced than that. And it turns out that truffles represent some of the most derived shapes of all the mushroom shapes. And it's because of environmental pressures that I'll talk about in a minute. So many their truffles and truffle-like fungi are found all through the tree. Any type of mushroom might evolve into a truffle under the right circumstances, but never the other way around. Once you're a truffle, that's kind of like where you are. It's really hard to evolve into a different kind of mushroom. So what happens, by theory, is imagine that you are growing in a place where there's climate change, and now instead of being a nice wet area that you liked, it's turned into the mushroom's going to die, go extinct. They're going to grow up, and the cap's going to open up and just get seared by that sun and killed. So they get afraid to open up and lose their moisture. And so they end up not actually opening up. And if they never open up, they don't need to have the shape of the gills. And so either the gill surface or the pored surface or whatever was inside just kind of turns into a ball of mush inside. And more and more and more, you can see them sort of just turning into blobs. So on the top left, we have it mostly looks like a mushroom. And on the bottom left, the top left has mostly looking like a mushroom. The bottom left, we have a truncocolumella, which is a pseuilus that turned into a truffle-like thing. You can see the bright yellow where the stem used to be. That's where the primitive stem was. And the other color is where the pore surface would have been. And here you can see a gill mushroom that's a little more on its way into a journey to becoming a puffball. You open it up, you can see what used to be the stem. And then the next step, well, they just turn into blobs, basically. Puffballs have a uniform marshmallow kind of texture inside, and that will eventually turn to powder, a cloud of spores. As you can see in the bottom left, a little hole might open up to let the spores out, and it's really fun to stomp on them and create this like cloud of smoke of spores. Others, earth balls, earth stars on the bottom left. That's kind of cool. They have this sort of extra layer of like a starfish that opens up to reveal the puffball inside. And on the bottom right, we have hygroscopic earth stars, which are really cool. They will actually, if they open up and they realize, oops, made a mistake. Forecast called for rain, but it didn't rain. They'll close back up again and wait for the better weather, and then they'll be able to reopen again, which is kind of cool. So then eventually they won't even bother to leave the ground because it might be such intense heat and sunlight that they would die just leaving the ground. So they stay where it's nice and cool and damp underground. But the problem is, how are you going to reproduce? How are you going to get your spores out there? So at the same time that the mushrooms are evolving into truffles, they're evolving strong odors to be able to be smelled from large distances by animals and dug up And so squirrels, some species of squirrels, truffles are practically 100% of their diet. They find them by smell and dig them up and eat them. And they run to another part of the forest and poop out the spores. And that's how truffles reproduce. They count on the fact that an animal is going to eat them. And they have to be found by an animal. So they have to evolve these scents. So that's why truffles are some of the more intensely smelling and flavored and most expensive and desired mushrooms, because they have the much more intense flavors, because they had to evolve them to be found even when they're underground. So if you look at the spores this is like a spores of a white truffle on the bottom left, they're spiny, kind of like a russula. Those spines detect the spores on its trip through the digestive system of a squirrel so that the spores are not killed by that time. So they have really, really tough spores. And we talked about spore pigments, how all of the dark spored mushrooms are related to each other. Why might a mushroom evolve a color to their spores? Well, that like sort of like the melanin, the pigment can protect them. So it's theorized that the thick walls, which a lot of your dung mushrooms, there's some psilocybes and um, uh, inky caps that grow in dung and have these thicker wall spores and dark colors to help them survive the trip through the digestive tract of an animal. So interesting fun fact, if you've wondered, you see a mushroom growing out of like some horse poop or something, how did it get there? Did it float around and a spore land on the poop, say, this is my substrate, I like it, and then grow up? No, the poop's not sticking around there for very long. It actually, typically what happens is the mushroom's been eaten by that animal, and then it's pooped out and the spore's already there, and it grows from it. That's how it happens. So true truffles are the ones you will find truffles in, as I said, in many, if not most, branches of the fungal tree of life. And the ones that we prefer the ones that have like onion and garlic flavors, whereas the animals aren't as fussy. The ones that have rotten cabbage and outhouse flavors, they just eat those too. But humans are a little fussier. And the ones that we eat are actually sort of closely related to morels. Maybe it's not a coincidence that our favorite tasting truffles are related to some of our favorite tasting mushrooms. So in this millions of years of evolution, the sort of final end product of millions of years of evolution is the ultimate shape that took the longest to evolve, this complicated stinkhorn that actually has like a knitted lattice work. No, weird kind of like bowling ball lattice structure there. Nope, it's a tiny blob. One of the most recently evolved that you could think of, one of the most advanced forms that a mushroom has taken is just a simple little blob. Centuries long mysteries have now been solved. With DNA, sometimes we already knew. The microscope taught us that Russula didn't belong with the other gill mushrooms. And the microscope taught us that the gill bolete was a relative of, confirmed that the gill bolete was a relative of the bolete. Sometimes we didn't notice, but we found a clue that we missed in hindsight. We found that DNA showed us that there's two kinds of inky cap. And we said, let's go look at that more closely. We said, oh, look at this. This one has so many gills that they're touching. That's different than all the ones that have few gills. That's how you can tell what family they're in. Like, that makes sense. Didn't think of that. But sometimes, and very often, we would never know that these things were different without DNA. We look at see things that are two different species, and we're struggling to find a way that a human can tell that they're different. There's a difference somewhere, but they can be really, really hard to find. The genetic diversity is extremely complicated. So in conclusion, some scientists have spent their entire lives trying to figure out how to classify mushrooms. They devoted their lives to it, and they never thought they'd have the answer. In Hessler and Smith's Waxy Cap book from 1963, they had this weird little waxy cap, and they were trying to decide, is it one of these kind or is it one of these kind? Because it kind of looked like both. They made a guess, and that's what they said. They admitted they were guessing, and they said, this matter will probably to continue to remain one of opinion as it has in the past. The end is not yet in sight. Well, the end is now in sight. With DNA, we will soon know how these mushrooms actually evolve back and forth and were related to each other. It is really fascinating to go back and see to go back to that book and look at that Wexy cap and see, did he make the right choice? Was it this one or this one? Was that gill polypore at the beginning? Is it really a gill mushroom or is it a polypore? Well, it's kind of more related to other things that are polypores and not gilled mushrooms. And I really wish that I could go back to these scientists and let them know, who spent their life's work trying to figure this out. We have some answers now that they would really love to have known, and I wish I could go back and tell them. All right. Thank you very much. That's my presentation. I'm here to take any questions.
Awesome. Wow, thank you so much.
You're very welcome.
There was a question early on when you were showing crusts, and it was would crust be considered lichen?
No, some lichens look like crusts. And lichen is a symbiotic relationship between an algae and a fungus. And they live together. And if they're not physically close enough to practically touch each other, they can't get the nutrition that they need, and they die. So some lichens are crusts. Many are. And so this is kind of a tricky question to answer. I guess it depends on your point of view. They are a crust shape, and they probably are one of the more primitive shapes. There's not a whole lot of variation of fungal shapes that came afterwards. So you can sort of think of lichens as crusts that way, the crustose ones. But most lichens are found in one class of ascos. But a lichen has evolved on other branches of the tree. There are actually two around the Pacific Northwest. We have two fairly common mushrooms. One is a little guild mushroom, and one is a little yellow club that are lichens that are used. So you actually see a mushroom sprout out of the lichen, and the mushroom is the fruiting body of that lichen. So in that sense, see, it's all how you think about it. In that sense, the lichen are not crust, because the fruiting body, which is what we're thinking of, the mushroom that comes out of it is not a crust, but not the reproducing fruiting body, but another part of the fruiting body is the crust. So it's different in that way.
But so you could kind of, like, you're calling crust a shape, or like
right.
An evolutionary shape,
meaningless in genetic or evolutionary terms. It's meaningless to say what the shape is. It's just turns out to be only something that a human will care about. Basically, these things are growing flat against the wood. It doesn't mean anything else about the mushroom than it was flat going against the piece of wood.
A way to describe it. One of the favorite things up here that we look for is chaga. And we learned that it's like, that is not the fruiting body, that the fruiting body is very, is this kind of hidden internal.
Chaga is a rare asexual fruiting of a polypore. Many ascos have asexual and sexual fruiting bodies, mushrooms that will come up that look different, like the carbon antlers that I showed back on the page, the little black coral that grows out, and they have these white tips. There are no pimples on it. Because it's the asexual stage. Rarely you will find a carbon antler with no white on it, pure black, and it'll have pimples on it. Go and look for one. Rarely you'll find it, and that is the same species. It's the sexual fruiting stage of it. So most polypores only have a sexual fruiting stage, but I believe, somebody correct me if I'm wrong, but chaga, there are no pores. You'll never see it doesn't look like a polypore. It's just a blob, because only the sexual fruiting stages will have the gills or will have the pores. So it's not that the chaga is the mycelium, it's this blob that's kind of an asexual fruiting body, which is very rare to see in comps. And that's one of the things that makes it so unusual. And we don't have chaga out here, and I'm jealous, because I would definitely have had chaga tea if that was an option for me instead of this one, because I think it's great. But unfortunately, it doesn't grow wild here. in the Northwest.
Um, Heather said, have you thought about writing a graphic illustration heavy history of mushrooms as a book? And she finds this super fascinating.
Oh, thank you. I appreciate that. I have not. I am more than busy making my DNA website right now. Which kind of demonstrates evolution because my DNA website, which I think somebody's posted, it starts basically with a tree, and you click on a branch, and it takes you to the page that describes them. So basically, you're looking at only the mushrooms, regardless of the shape, that are genetically related to each other. So it kind of tells the story by browsing through that tree and seeing what things that you see at the different points of the tree.
Very cool. I posted that link again. And anyone, please, if you'd rather unmute to ask a question, please feel free to do so. John and Claudette said it's super fascinating, but DNA is still driving them crazy. I think some of us wonder, do you think there are anything about the process, the DNA, or the way that it's being done that it could be found that it's not the whole story?
Good question. I mean, there's a little bit of hubris there saying now we have the full story because we looked as closely as we could. We thought that's all we can do. We invented the microscope. We said, oh, well, now we can see the tiny things. I'm sure that's all we can do. We have DNA. And now I'm saying now I'm sure that's all we can do. So I have to have a little bit of humility to say you never know. But it does really, since it is showing us the story of being able to see how one changed into each other and the whole process, not just the end points. That is different and a part of the story that's never been able to been told before. So I can believe that it's at least close to the end of the story, if that makes sense.
Totally. Yeah. Any other questions for Danny? Is anything that he showed you you've never seen before, or something that really blew your mind? And it's like you said, we're all so interested in the shape and naming things, and just having our eyes tell us the story.
Yeah, what I think is really interesting when I was first learning before DNA, how meaningless identifying mushrooms now feels in a way. Like you would go to the key and you would try to figure out what you have, right? And it was this white spored gill mushroom. It had decurrent gills running down the stem like chanterelle and kind of funnel shaped. And it would say that's a clitosoby. And then you would think, great, I identified this mushroom. I realized I found a clitosoby. And they say, so what does that mean? The definition of a like I said, my white spored recurrent gilled mushroom is a clitosoby. And somebody says, what's a clitosoby? It's a white spored recurrent gilled mushroom. Like it's tautological. You didn't learn anything. You just said, what does it look like? And I gave you a name that described that shape. You didn't learn anything new about it. You don't know what its ecology is. You don't know what it's related to. And it turns out I choose clitosoby because it's been split into a dozen different families, dozens of different genera, because that shape has evolved so many different times. You really don't know anything about it by knowing that it used to be called a clitosoby. So only once you get DNA into the picture can you say, I put a name on it and that means something. I can now go say look up clitosoby and say, oh, that means it has this ecology, maybe it has these poisons or not these poisons. Whereas before, all you could do is if you told somebody you have a clitosoby, they could go backwards and say, oh, I see you found something that was white and had gills running down the spine. That's all you could say about it, is to go back to the original thing that you said. It wouldn't teach you anything new. I don't know if that makes sense, but that's kind of my understanding of why it's worth going through the name changes and understanding what's more, even though it's much more complicated to learn now, why it's important, because only now can you learn things that are going to be meaningful.
Awesome. Yeah, before it seems like a lot of it it was developing a language so that you knew that when you were talking to someone, you're talking about the same thing. But if you all, like you said, if you all knew the names, but that's all you knew, you're just saying names back and forth to each other. You're not
Right.
getting any
Yeah.
deeper. Um,
Yeah.
there's some other questions here.
Yeah. You could think of it as shorthand, like it's just quicker to say clitosomy instead of mushroom with white spores with the gills running down the stem and a funnel shape on the top. It's just a shorthand way of saying that. Think of it that way.
And
Yeah.
then what? Yeah, yeah.
Right.
um, so I think Dan, cool. What are the most prolific mushrooms across the biggest habitat? North and south. Dan, is it Dan? Let me know if we're understanding your question. Um, so I guess, yeah, the biggest and
Well,
the biggest habitat and the most prolific mushroom.
yeah, there's some mushrooms are really specific. You'll only find them under one tree. Some are sort of generalist. They like many, many different conifers, or they might only like hardwoods. Some are super generalist and they don't care at all about the tree in some ways. So there's definitely things that range across larger habitats. I know that boletes and amanitas in the east are there's much more numerous than in the west. Like you have as many genera of boletes as we have species in the west, practically. Although they're they're worldwide. Um, so I guess the way I would think about it is that there are definitely different mushrooms are more or less picky about their ecological niche. And some things like ramarias, the colorful corals, are tree specific. There is practically zero overlap between the species found in Europe and the species found out here because the trees are different. They are specific to basically one specific tree.
Cool. Uh, Michelle is asking, what do you think DNA identification will do for the future of foraging? Will we still make identification books, given it's near impossible to be accurate for the DNA results?
Oh yeah. I think they will always be they'll just be it just means that you're going to have to update your book if you want the current name. And you don't even have to. I mean, the old names will always be valid legal names for them. It won't be as informative. You'll like call it a clitosomy, such and such. Go look up in the internet that species, and it'll say, "Hey, it's been renamed and has a new name." You didn't have to care, but you can find the new name from the old name and then find out what that means about it and learn more about it if you want to. But it doesn't mean that you have to learn the new name. So don't despair. Nobody's going to it's kind of like they retire all their jersey numbers. They're never going to reuse a name of an old mushroom for something new. So that name that you knew years and years ago will always mean that mushroom. Now, sometimes we find that there's more than one species that look the same. So that old name could map onto two or three different new names. But they'd all be very closely related and share almost all properties you could think of together. So that's not quite as you don't need to worry so much about that, I would think.
Gotcha. That's interesting to know that once a name was a name, it's not going to revert to something else. It has to become
Right.
something new. So if you did have an older book, you could trace that history. That's a
Yes.
really good point. Daniela is saying, what can we do to support the evolution of your work?
Oh, thank you. The thing that's hard is now that DNA sequencing is so easy, we get thousands of sequences come back, and few people are trained to know what to do with that sequence and what does it mean. Fundus, which is a national non-profit, um, has Hartsinger is the leader of a DNA sequence analysis team. You can contact him or anybody at Fundis, Fundis, can put you in touch. And where they've developed training programs and ways for people to get involved and look at the DNA and help out. And it's really fun. It's cutting edge stuff.
Oh. Oh, that's cool to know. Yeah, I think as amateur mycologists, we sometimes think of ourselves as, the hands and feet on the ground, like at the NAMA foray and stuff. But to be able to actually participate at a data level, I bet there is a lot of
There's a lot of use for that.
work to
Yeah.
do. Very cool.
Yeah.
Randy says, for your project, do you do nanopore sequencing? Has nanopore become more economical for sequencing relatively small numbers of samples at a time?
It certainly has. There's several different ways that you can DNA sequence something. I have another talk that goes through how each of those work. But nanopore sequencing is much cheaper. You basically do 1,000 things at a time instead of 1 to 24 to 96 at a time. And it is much cheaper per. It's If you do that many at a time, you have to find the right primers and the right chemicals that work across everything. So it sometimes you have to do the old school Sanger way. But for the vast majority of things, nanopore has been a game changer. It has really brought the price down from $30 to $40 each to less than $10 each, or to just a few dollars a piece for sequencing. It has been revolutionary.
Wow. And John,
And And you can do it at home on your table. It's a USB device. So it used to be that you could buy a centrifuge and a bunch of chemicals, and at home you could stir them all up and mix them all up to get everything ready, and then you'd send it to a lab to do the DNA sequencing. You could do the first half of the job at home. But the nanopore thing is a USB device that attaches to your laptop. So now you can just sit at your kitchen table and do the complete DNA sequencing without a lab. It's a home lab kit.
wow. Wow.
Like
And
$1,000 or a couple $1,000 to get the equipment for the first half, and then it will cost you like $1,000 for each 1,000 things you want to sequence. So like $1 a piece is all. When I say like $5 to $10, I mean to send it to a lab. But if you want to do it yourself, it's $1 a piece or less, which is still a lot. It's still $1,000, but you'll get 1,000 sequences. That's like so much better than 30, 40 bucks a piece.
well, especially for like a club who, over the course of a season is going to find hundreds or
Yeah.
thousands of specimens and wants to kind of learn that. No, that's
Yeah.
really something cool to think about.
Yeah.
So
You
John
could get
Cole
one, you could get a club one.
yeah. Is there any indication that Graphola frondosa is actually more than one thing? I don't know if you guys are studying those. I don't think you have them on the West
We
Coast.
don't. We rumor that like somebody claims they can find one every 5 years, and maybe they can, but it's certainly pretty rare. It is, I don't know specifically, but I know a big reason of why I say the biodiversity is so big, of why there's so many more species, is that what look like one species is turning out to be several. And you look a little closer and you see, oh yeah, there were differences. So I don't know yet if that's true with Graphola. I think being kind of a monotypic genus where everybody was calling anything in that genus the same thing, at least geographically, I'm sure there's more than one species. I would guess there is. That's the typical story for something like that, is that it will turn out to be more than one species, but I can't say that for sure from specific knowledge of that.
John, did you want to unmute? I know I've thought that same thing. I've seen, again, this is just what I've seen, but things look, taste, feel sometimes different, and I've thought that same thing that
It is so prone to variations.
Mhm.
And there's some that come early in the season and some that come later in the season. And, they all
Sounds
seem to
suspicious to
be,
me.
we see them so much around here. That's why we particularly notice them.
Sounds suspicious to me. Yeah.
Yes, right.
Yeah. You could take
Um,
one of each of them, and this is what's great about citizen scientists club members can meaningfully contribute to the science, because I don't know if anybody has, but like at least a few years ago when nobody has checked it out, you could have been the person who've collected one from each season and each different thing, sequence them and discover if they're the same or not. Like you can make meaningful contributions like that to the field. That's what makes mycology so fun.
we noticed too that a lot of times it's something, or you would think it's something really obscure, really unique. It's like it has to be such a special thing to study it, when in reality, some of the more common things that we already kind of know, we think we know, we don't go back and ask because we're kind of thinking, oh, we need to look at the very unique thing and study it, or the thing that feels rare versus the very common thing, and study that.
Yeah, just in the past few years is where we've discovered that a surprisingly large percentage of the things that people have been collecting all their lives didn't have a name. I don't know, 10, 20% at least of the mushrooms that you think you know have probably changed names lately or still need to. It's not just the rare ones. Like what it turns out we're starting with a much lower set of knowledge than we thought we were. And now we're starting to work our way through it, but it's going to take years.
There's a if people want to look it up, there's a really well-known, it was Tom Volk down at La Crosse, which is really close to us. Had a student that did the chanterelle study and like found 12 different species in one small area that they were doing a foray. And to us, we're just like, chanterelle, I know what it is, I can eat it. So again, something that's just all there right next to each other, and because we have a name, we feel like we've learned all there is to know. I'm interested in that when you get past a name.
Yeah,
And,
our West Coast chanterelles a decade ago or so got split into 5 different things. Yeah.
Wow.
yeah, and in his case, his student found that there was a pair that they could not morphologically see any difference microscopically, but the DNA said they were different.
Oh, this is what DNA
Two different
analysis is interesting because
two different chanterelles, but when they went back and tried to figure out what they missed, they couldn't figure it out. They said it's the only way they could
yeah,
tell.
that's
Interesting.
cool.
So
Happens a lot.
asking, how fast is DNA analysis on individual samples? Are we talking hours, days, weeks?
Pretty fast because there's tools now that once it's out, the computer blasts it and it says, here's the things that match. Now, the problem is GenBank, which is the repository for the millions and billions of sequences that people have done, most of them are misidentified. So you don't know what you have unless you have cultivated your own private database of sequences that you trust from people that you trust who are experts in that particular genus. So I've created my own trusted database that I blast against so I can see if it matches one of those or if it's different. So if you get a match to something that you trust, you'll know right away what it is. You still have to look at right, and see where all the differences are. But you can do it in just a few minutes. But if it's something that doesn't match any sequence seen before, then it's going to take time. Then you have to go back to the raw files and say, was this a mistake? Is this actually something went wrong and this is not actually the DNA sequence? Then you have to investigate that. Then you say, okay, it is real, but it's different. So now you have to decide how different it is, and is this different enough to be a new species, and what is it? And it can take upwards of an hour or so for the really strange ones. But in just a minute or two, if it's something common and a good match, you can usually know what it is.
Wow. So is there any in the history of the recent history of people doing DNA analysis on things, has there been any change in how they're doing it so that there's, like you were saying, there's different data sets where it's like one data set's looking at one thing, another's looking at another? Like, would you say that there's a certain year or time where all of a sudden it was like, now we got it down, I feel good about all this data?
I would say every few months it's changed and improved, and no one's willing to say they've got it down yet. It's that new. Yep.
Wow, exciting. Cool.
Yeah.
And then Erica, it says, I recently returned from your lovely region. on spring break and found a lichen agaricus, Lichen umbellifera, umbellifera.
Yeah, that's the little guild mushroom that I mentioned. That's actually a lichen. It's a lichen that sprouts a guild mushroom.
So she says, on
It's
the website project you discussed, DNA might be reclassified for that. I'm still new and learning, but a fruiting lichen blew my mind. Yeah.
yeah.
How
Yeah,
to how
it's related to the waxy caps. It's in that family. I mean, it's kind of colorful. It's got these little like yellowish and orangish colors, but you wouldn't know. But that's where it just happens to be a pretty close relative of the waxy caps.
how does that work in a lichen that way? Okay, so where's the genetics of the algae in that whole?
Yeah, well, the mushroom is the fruiting. It isn't actually a cross, like they aren't mating, but they're living in
No,
close
no, right.
proximity. So if you just get the mushroom and sequence it, you're only going to find, I believe, the fungal
Okay,
DNA. You're
okay.
not going to find any algae DNA.
Mm. And then once the fungal spores start growing, then the algae finds them.
Yeah, or they'll have to find each other, or the spores will have
Right,
to
right,
find the algae in order to, in order to actually
right.
germinate, perhaps.
Yeah. Hmm.
Well, this has been some great discussion too, even after all of your lovely pictures and description. Oh, I thought of one more question. When you were talking about
Yeah.
the inky cap, that was super fascinating. So do you think as a part of the evolution, the growing of or the having more gills was kind of like in that same vein as the more surface area, more spores? It was sort of like more is more, but then it um
Got
became
too
a
carried
detriment.
away.
Yeah.
Yeah, maybe, because the more gills, the more spores, the better. So there could be some pressure to make more and more, and then it reached a point where they couldn't fit in, it didn't work. And many times that would happen and they would die off. So you would not get the mushroom to evolve gills that close together. It wasn't working. The ones that did would just die off. But then once, by a miracle, at the same time that happened, it evolved the way to turn into ink, and that one didn't need to die off. And then it became an inky cap. That's the theory.
Interesting. Interesting. Well, thank you again. I'll do one last
You're
call
very welcome.
for
This was really fun.
any
Yeah,
questions or announcements from anyone, but yeah, we'd love to keep following your pages. Thank you for all the work you're doing for mycology. It's
you're welcome.
Thanks. It's been really interesting.
Yeah, it's been really fun to be here. Thank you.